My name is Shreyas and I'll be explaining you what phosphorylation is and how it is done. So, what is phosphorylation? So, as you know, like our biological systems are extremely complex. Like, there is stuff constantly being done, there is stuff constantly being regulated, and all on its own. So yeah, partially all on its own sort of. It's like an automatic system. We don't have to give like signals every time that manually. Oh yeah, get this done, get this done, get this done. There is so much stuff happening. We don't even know about it. So all that is regulated by very complex mechanisms, and proteins are like a very crucial part of these mechanisms. So what phosphorylation is basically is the transfer of a phosphate group onto a protein. So why is this done? This is done. Okay, this. Yeah, I'll just erase this. So phosphorylation is basically, as I said, the transfer of a phosphate group onto a protein, and this is done one to activate the protein or deactivate it for that matter. I'll explain this. So activate how? So if when a phosphate group attaches to a protein, it changes the protein's conformation. Um, this is just related to the way like how the amino acids are attracted to each other and how the proteins fold. So yeah, basically the conformation changes. On this conformation change, it can either become active in some activity that is going on, it can also become deactive, and basically the function of the protein itself can change greatly. So yeah, that is how a phosphate group can deactivate or activate proteins. Um, so another thing that can happen is protein signaling. So proteins have this complex signaling mechanism, and it's how they communicate with each other. So a phosphate group, when it attaches to a protein, it attracts other proteins. So when other proteins are attracted, they're sort of like communicating between each other, and this chain of attraction is like sort of carried forward. So yeah, this is a very important thing to be able to regulate different biological mechanisms. Uh, yeah, so that is why phosphorylation is important. So how does this come about? Who does phosphorylation? So there are proteins called kinases. These kinases are responsible for bringing about phosphorylation. So what they do is uh, okay. So we talked about how phosphorylation requires a phosphate to be attached onto the protein. So obviously this requires some phosphate source, right? That source is ATP, which is adenosine triphosphate. So this molecule right here contains three phosphate groups, the alpha, beta, and gamma phosphates. Out of them, the gamma phosphate, which is like the end phosphate, is the one that is transferred onto the protein. So a kinase picks off this gamma phosphate and transfers that to the protein. So obviously, in a regulatory mechanism, if you have something that is getting phosphorylation done, there should also be something that gets sort of dephosphorylation done. That is, it something that picks off the protein, and those substances are called phosphatases. So what phosphatases do is they pick off this phosphate group from the protein. And yeah, this is also obviously important for the regulatory mechanism. So let's talk about how kinases actually do the Okay, right. So before that, let's bring about an important point. That is, not all proteins undergo phosphorylation. Only some proteins, and I'll name them: tyrosine, serine, and threonine. These proteins mainly undergo phosphorylation. There are some other proteins which also undergo phosphorylation, but we won't talk about them because they don't always undergo it. There are some specific conditions that need to be there, and like basically they undergo the process only under like very specific conditions. So yeah, we won't talk about them. So about tyrosine and serine and threonine, not all. So kinases come in like sort of different variants, and not all kinases are able to like phosphorylate all three. So there are some kinases. There is like, so basically there is kinase category number 1 that is able to phosphorylate tyrosine. Then there is kinase category number 2, which is able to phosphorylate serine and threonine. So yeah, basically any kinase that is able to phosphorylate serine is also able to phosphorylate threonine. And then there is kinase category number 3, which is able to phosphorylate all three of them. So that is how kinases are classified, based on serine, threonine, and tyrosine. Yeah, so now let's come to how a kinase actually transfers the phosphate group from an ATP to the substrate protein. So there are three things to consider in the topic that we're discussing about. That is the ATP binding. These are the three main aspects to a kinase. One is the ATP binding, the substrate binding, and the phosphotransfer. So initially, there is like an ATP molecule. A kinase will come and trap that ATP. So this is that ATP, the pink color thing. It is trapped inside the kinase. Now so how the kinase does it? The kinase has a glycine-rich loop, which basically extends over the ATP and sort of traps it inside. Yeah. So once the ATP is now trapped inside. There is also a conserved lysine group in the kinase that will hold on to the alpha and beta phosphates of the ATP. So now the gamma phosphate is the one that is supposed to leave, and that is not bound. So yeah, this is kind of designed in a very smart manner. The final part is the catalytic aspartate part of the kinase, which is the one that interacts with the protein substrate, the one that is supposed to be phosphorylated. So this is the group that is responsible to actually remove the phosphate from the ATP and then transfer it to the protein substrate. So this is how phosphorylation is done by kinases. So yeah, that is it about kinases and phosphorylation. Keep enjoying science. Goodbye for now.